Boa tarde, pessoal! Estamos na live finalmente. Sejam muito bem-vindos a mais um episódio do LEABTalks. Eu sou a Fernanda e já começo agradecendo a todos pela presença. Hoje nós contamos com a presença do chefe do nosso laboratório, o professor Dr. Danilo Vicensotto, e também do Dr. Pedro da Glória, que é docente do programa de Anthropologia da Universidade Federal do Pará e pesquisador colaborador do LEAB. Nós gostaríamos também de estender nosso agradecimento a Daniela Oliveira, que é membro do laboratório e foi parte essencial desse episódio de hoje, mas infelizmente não pôde estar com a gente nessa live. Bom, lembrando vocês, o LEABTalks é um dos projetos de divulgação científica do laboratório, destinado à organização, promoção e divulgação dos mais variados temas científicos através de palestras com cientistas e pesquisadores. Para mais informações sobre esse projeto, a gente vai deixar na descrição do vídeo depois os links de acesso ao site do LEABTalks e as redes sociais do LEAB também. Nesse terceiro episódio, nós temos o prazer de receber a doutora Julia Gamble, é professora associada do Departamento de Antropologia da Universidade de Manitoba, no Canadá. A doutora Gamble é graduada em Antropologia pela Universidade de Winnipeg, mestre em Antropologia pela Universidade de Sheffield e doutora também em Antropologia pela Universidade de Manitoba. Sua pesquisa envolve a investigação sobre a saúde no passado, buscando compreender as interações iniciais da vida e a saúde posterior, através, por exemplo, da análise do esmalte dentário e resquícios ósseos humanos. São, então, suas grandes áreas de estudo a bioarqueologia, antropologia dentária, paleopatologia e osteologia humana. E hoje, a sua apresentação será sobre a intersecção da bioarqueologia e antropologia dentária, aprendendo sobre populações passadas através de uma abordagem multifacetada à saúde e ao estresse. Então, lembrando que ao final da apresentação da doutora Gamble, nós vamos abrir para as perguntas, mas já podem ficar à vontade para ir mandando suas perguntas no chat do YouTube. Okay, Dr. Gamble, you can enter now. Thank you so much for meeting us today. It's a pleasure to have you here.
Hi, thank you so much for having me. I'm very glad to be here.
It's our pleasure. Okay, so now we can close our cameras, and after the vignette, Dr. Gamble, please feel free to start.
So thank you for the introduction. I'm very glad to be here, and I'm going to be talking to you today about bioarchaeology and dental anthropology and what we can learn from kind of a multifaceted approach in terms of past human health. The populations that I work with are both in Europe. I've done a fair bit of work in Denmark, and most of what I'll be talking to you today about will be those populations. And I'm going to finish off by talking a little bit more about some more recent work that I'm doing in Austria, and really that is a very much beginning stage in terms of trying to understand a new population. In Denmark, where I've been working for quite a few years, we are interested in a number of questions about human health and stress and responses to environment and the circumstances people were experiencing. So I'm interested in how people experienced early life, and in particular about how their early life experiences impacted potentially their later life. And I look at that in terms of aspects like age at death and any other pathology, and I'll talk more about that in a minute. The context in which the people I study lived also involved a range of circumstances, including factors like famine that I'll talk about in a minute, and the Black Death. So we're also interested in how these factors might have influenced them. And finally, we're interested in pulling this together to a broad picture of what life might have looked like for medieval people in Denmark. So the area of focus again is in Denmark, and we are going to be looking at populations kind of throughout this region. These are the main sites that we're going to focus on, and so you can see that they are spread across Denmark. And what we're seeing in medieval Denmark is a pattern of changing population density and settlement patterns. There is a shift towards increasing urbanization through the late medieval period, with the focus being from the roughly 11th century through to the 16th centuries. During this period, there are climatic fluctuations, which you may have heard about before. There's what's been termed the Little Ice Age, which lasted from about 1300 AD to 1860. And the Little Ice Age is a little bit more contentious. When it starts and its duration, the period it spans, depends a little bit on who you're talking to and what measures of climate you are referring to. Whether you are using, for example, glacial records or climatological patterns. So glaciation is a little bit different from climate, although of course they're connected. So there's a whole other literature about when the Little Ice Age occurred. But what we're seeing here with Denmark and with Europe is periods during which there might have been some very tough winters, potentially a few rough growing seasons. And when you're dealing with a population that is agricultural, there is a limited capacity potentially to kind of come back if there's been a difficult season. So you might manage to save your seeds for a year. But if you have two rough seasons, that's going to complicate things even further. And so what we might have been seeing during this period are a few seasons, a few years that were rough enough that they caused issues for the population. So we're interested in how that affected population frailty and resiliency. And then the second phase is the late medieval agrarian crisis. This is kind of tied into the Little Ice Age. It is a period which saw some difficult times, including relating to crop growth and also things like potential pestilence amongst cattle. So the Great Famine occurred from 1315 to 1322, and this was a particularly rough period that we see. And during this period, we also got cattle dying with a bovid pestilence. So these were probably some very difficult times. And then a few years later, 1349 roughly to 1350 entry, we get the appearance of a disease which you no doubt have heard about, the Black Death. And there's still a lot of work being done to figure out what this disease was in terms of why it affected the population so severely, but it likely had fairly serious impacts that we still see today. Now, I'm interested in understanding what these patterns meant for human health, how resilient people were, and what it meant across the life course. And to do that, I look at various aspects of the human skeleton for people who lived during this period. The human skeletal remains can provide a lot of context about health through the life course. And my initial focus was on adults. So I was studying people who it was from their bones, relatively straightforward to identify whether they were male or female. And we could get an idea of age. Now, My main focus, though, was at trying to connect the different points in their life. And for that, teeth are really useful because teeth only grow during childhood. In particular, dental enamel completes formation with different teeth completing formation at different points in time. And dental enamel is also very hard and therefore fairly likely to survive. And it captures a record of growth, therefore restricted to the childhood period. That growth is apparent as what we call incremental growth markers. So dental enamel grows kind of like tree rings, except that it's each growth line represents a shorter period. So tree rings grow annually, and what we have in teeth are lines that represent roughly daily and then roughly weekly markers. And if you get disruption in growth, you can get a permanent mark indicating that in the tooth enamel. So I was initially very interested in looking at these incremental markers of growth and in trying to understand what that meant in terms of childhood responses to stress, and also then how that affected people's resiliency throughout life and people's survivorship and response to disease. So this ties in with a framework known as the Developmental Origins of Health and Disease Hypothesis, the DOHAD framework, and it speaks to the connections between early life experiences and later life health. So incremental dental microstructures can be seen on both the surface and the internal parts of the teeth, and I'm going to show you that more in just a second. The teeth can, however, show you a lot more information. You can also see incremental markers of growth in other dental structures like dentin and potentially even cementum, although they represent slightly different things. So that is an area that I will be exploring. I haven't yet. But teeth also can tell us about an individual's sex. And so I'll be talking about proteomic-based approaches to sex estimation from a protein in dental enamel. They also provide amazing repositories for DNA. They're kind of little time capsules. And so we've been doing ancient DNA work to try to get at pathogens. And finally, they can tell us about diet, both in terms of isotopes and in terms of dental calculus that gets preserved on teeth. And so these are aspects that we kind of keep at the back of study. And I'll talk more about how this all comes together at the end in terms of trying to really extract as much information as possible from very small samples effectively in the form of a tooth. But all of this gets tied together with something that's very important to archaeologists, which is the concept of context. So understanding what was in the environment of people as they were living. And part of that is understanding the disease landscape, and that's where I will talk about Black Death. All right, so if you zoom in on a tooth and look really closely at the dental enamel, this is what you will see. So the surface of a tooth, this is done with a laser microscope, a Lex laser microscope, has all of these lines going across it. You can see that in the top image going from top to bottom. And these are parenchyma. They are waveform growth structures, and they form at roughly 7-day periods, although it ranges in humans. Somebody might have a 7-day cycle between lines, another might be a 15-day cycle between lines, for example. And sometimes you get an irregular spacing between them. As you can see, some of them are more pronounced than others, and that indicates that the person underwent some change in their growth relating to systemic stress, so something that was affecting their body at that point in time. And because they form very regularly, we can theoretically figure out exactly what age they were when that stress marker occurred. An issue with these surface markers is, of course, people eat, and when they eat, their teeth get worn down, and they get abrasion and scratches across them that can obliterate the stress markers. Another issue is that the very tip of the tooth, these markers never come to the surface, so you wouldn't see it. Now, to get at this in more detail, to get past these problems, one thing we can do is thin section the tooth, and that is what you see in the bottom image. That is actually a cross-section of a tooth where you can see these growth lines extending from the surface, which runs along towards the top of the image, down towards the dentin, effectively in the pulp chamber. So when you see a particularly pronounced line, that's an accentuated stria retzius, and that indicates that there is a disruption in growth. So I looked at teeth on both of these levels, both in terms of the surface and in terms of the internal. And I looked at this in relation to survivorship, the age at death initially for adults, because I was interested in seeing whether there would be a difference in males and females. And at this point, we weren't able to really identify male or female from children. Children's bones do not form very clear sex indicators prior to puberty. So when I do this and run what we call these survival analyses, there was an interesting pattern I didn't actually expect, and that is that males and females show different patterns in survivorship depending on the amount of stress shown in their teeth. The surface scans showed that males with higher amounts of defects didn't survive as long, but females, we're not seeing really an effect at all. When we actually look at the internal pattern, we continue to see these patterns, except for females, actually, there seems to be a suggestion that not only do females with more stress not see a negative impact on survivorship, but they may see a positive impact. They may actually survive longer if they have more stress. And I'm not entirely sure what this means. I still am not sure what this means. The only thing I can really say is it does seem to consistently suggest a difference between males and females in how their survivorship is impacted by early life stress. And I won't go farther than that, but it's quite tantalizing. So the question of why this is happening leads to two primary hypotheses. The first possibility is a sociocultural explanation for these populations. Maybe there was a difference in how males and females were being treated in childhood, and this affected their resiliency later in life. So there could be something sociocultural happening. Alternatively, it's possible that we have a biological explanation, that there is some fundamental difference between males and females on a biological level that is affecting their resiliency in relation to early life stress. Now, this is difficult to get at when you do not know whether when you can't look at children in terms of sex. So without understanding, being able to look at male or female children, it's hard to tease this apart. And the historical records don't give us much information either, because first, females tend to be underrepresented in historical documents, and second, children tend to be underrepresented. So historical documents from the medieval period do not tend to tell us much about how children are raised or how males or females might have been treated differently during childhood. So it's a difficult one to get at. And this is where we get into more recent methods for identifying sex for children. Now, we would have been able to do DNA testing and to try to determine sex that way, referring specifically from a biological binary sex estimation. That can be challenging. It can be expensive. It is obviously destructive. So a few years ago, we had some amazing work. Parker and Stewart are at the front of moving this forward. And with those early publications, I decided to see what we could do with the medieval Danish populations. And so we have done sampling on teeth for something called amelogenin, which is a protein that it's a main protein in dental enamel. And so it's quite common in dental enamel, and dental enamel being quite hard preserves it very well. And This protein is coded on the XY chromosomes, so you get a slightly different shape for the protein if there is a Y chromosome present, indicating that you have a male. So we decided to try this on a fairly large sample of medieval Danes, and we validated it using a modern papillonation of known sex. And we were able to do this successfully on most samples, even when there appeared visually to be poor preservation. And this meant that we got quite a clear separation between the females and the males when we run it through a mass spectrometer. And we were able to attain this for children and some deciduous teeth, meaning some baby teeth. So this was really, really useful. And it means that we can start looking at these stress markers in teeth in relation to child survivorship. So we have the adult survival curves. The lower ones are the adult ones you have seen already. So you can see that male survivorship, severe impacts on the dental growth result in reduced survivorship. Now, what then is going to happen with the children? And when we do this, we see a similar pattern. So again, the green line represents severe effects on enamel. This is just the surface. We haven't run anything in terms of the internal structures yet. I am very slow to cut teeth because it's a destructive process. Amelogenin, incidentally, is not very invasive. It's a bit of a dip in hydrochloric acid. It hardly changes the appearance of the tooth at all. It demineralizes it a little bit, but it's not very destructive. So we haven't gone to the next step of actually cutting these teeth. But what we do see is that the males with severe stress in childhood also died younger. With females, we are not seeing that effect. If anything, the green line is higher, so we're seeing potentially a slight positive effect, but it's not significant. So there seems to persist a difference in survivorship during childhood between males and females. Now, this still doesn't help us answer completely the sociocultural versus the biological question. But I think it is probably speaking to something that is very innate, certainly within this society between males and females. And further steps would be to consider whether we see a similar effect in other populations that might be biologically or genetically different and culturally different. So there's more testing needing to start to control. But this leads to trying to add further context as well, because I'm interested in what people are actually experiencing, what might have been causing stress, what might have been affecting resiliency. And one of the things that we know happened during this period was the Black Death pandemic. So I'm going to show some pictures of rats and bugs, because we're going to get into the Black Death a little bit for people who are less familiar with it. So the Black Death has often been attributed to bubonic plague or to plague, which is caused by the infectious agent Yersinia pestis. And there's a lot of contention still around the complexity of why it was so severe, but we do know a lot about how plague actually spreads now. So this is a pathogen, a bacteria that has a natural cycle of moving between fleas and some sort of rodent. Rats are the most commonly known. Black rats are known from historic plague, but we know now that other things can be involved in this framework, including things like gerbils. So what happens is the flea bites an infected animal and ingests the bacteria, and those bacteria kind of spread in its throat and make it so that it can't properly swallow. And then when it bites again, it regurgitates and spreads the disease further. So this is the natural pattern. That's at least for bubonic plague. It can also, if it gets into the bloodstream, become septicemic. If it gets into the lungs, it can be pneumonic. And sometimes humans get into the mix. They get bitten by a flea, and they can suffer from plague. Now, so this has been proposed for a number of years as a primary cause of the Black Death episode, and there's been a lot of ancient DNA work to try to identify it in past populations. And I, coming out of my PhD a number of years ago, was very interested in understanding not just this disease, but what other diseases were affecting the population that I was studying. And so I reached out to folks at McMaster with Hendrik Poyner, and we developed a project. And we're still working on the broader context, but we do know something now about plague in Denmark. So what we do is we sample teeth, and I will caveat this. I am not an ancient DNA expert. I'm a bioarchaeologist, so my amazing colleagues at McMaster and farther afield are really at the forefront of engineering this side. But what they do there is they cut the tooth, and then they extract genetic material from the pulp chamber. And this takes place at the McMaster Ancient DNA Center. And then they start to process to look for fragments of DNA. And we specifically are interested in pathogen DNA, as I've mentioned. So we sampled a number of sites. What we know from historical documentation in Denmark is that the first appearance of plague is probably somewhere around 1348, actually in Oslo, in Norway. And then by 1349, there seems to be evidence that it might have struck Denmark in a couple of locations, in Halmstad, and then over actually in Ribe. We've got some politics with Sweden and Denmark. I'm referring this as kind of the Denmark contingent here. And then in 1350, by 1350, we have it in the rest of Denmark, okay? And so we have sampled sites in the rest of Denmark to try to understand whether plague is present during the Black Death episode. So what we have is a successful identification of plague in Denmark that doesn't happen before the mid-14th century. So it suggests that it did enter Denmark with this Black Death episode. And then it continues in Denmark. And it's very closely related to the pathogen genetics that are elsewhere in Europe, suggesting that there is kind of a reintroduction. We're probably not getting continued evolution in Denmark. So we sampled 298 people in the end over 800 years, and these represent 13 different sites. And we found plague from the 14th to the 17th centuries. And this kind of shows you on the right where we found plague pathogens sufficient enough to basically extend and get a genome out. So that represents about 9 people. 9 plague genomes we were able to do phylogenetic analysis on. And it shows you kind of how they fit in relation to other plague genomes. And so we see that the earlier ones appeared in Ribe, which is consistent with the historical evidence. And those are also genetically similar to that Black Death episode genomes that have been identified elsewhere. And we then see other genomes appearing, including the more recent ones being related to modern plague. And finally, the last one appears in Falberg, right up here. And that seems to be the last episode of plague in Denmark, and that is also consistent with the historical record. So this is from a publication that came out just a few months ago, Catherine Eaton and colleagues. So it's a huge team of amazing researchers that we're finally getting to see what is happening with plague in Denmark and to see the connections to the broader European context. So we know now that we've got plague in Denmark. It coincides with the Black Death literature, and it's quite interesting that it was affecting some of the people that were included in the early PhD sample that I looked at, and also affecting both urban and rural populations. And I'd like to dive into one case study here, because part of what I do is understanding on the population level, but sometimes looking closer at individuals and creating a bit of a kind of an osteobiography of them, we would say, can be quite interesting and can speak to how different forms of evidence can give us insight into the lives of people who lived in the past. So this is a site known as Tirrup. It's a rural cemetery that's been studied really quite extensively. It lasted from about 1100 to 1350. Its demise has not been tied to the Black Death. We may be seeing a connection with the Little Ice Age or the Agrarian Crisis, but this is considered to be, it's a completely excavated cemetery, and it's considered to be kind of tied to that period. So it's really valuable, and we know quite a bit about it. And it was one of the populations that we sampled for the Black Death study and the pathogen study in general. And I will caveat that the next slide will contain pictures of human skeletal remains. So this is just something that I like to mention in case people don't really like to see human skeletal remains. So those will be up on the next slide. So one person who is quite interesting is this young woman here. She was a female who lived in or at least was buried in Tirrup and died between 25 and 30 years of age. She does have enamel defects. You can see with a red arrow just visually here. That occurred somewhere around 5 years, at 5 years of age, so when she was a child. Her skeleton looks pretty healthy if we're talking about osteological changes and paleopathological analysis. There's nothing really that stands out other than she has a bit of enamel defect, and there are a few places where if you look really closely, there's maybe some inflammatory response. Now, interestingly, she does come up as plague positive. So this person did have plague when she died. Whether that led to her death or not, we can't really say. But she also has some suggestion of other co-infections. We're not entirely sure what, but they're tied into the mycobacterial complex. So whether it's leprosy or tuberculosis, more work is needed. Again, this is work that's being done in conjunction with McMaster and Catherine Eaton's work, Dr. Eaton now. So this is just piecing it all together, that the paleopathology does have some indicators that are in the ribs that normally a paleopathologist might see but wouldn't be able to say was tuberculosis. There are maybe some indicators in the feet that we wouldn't really be able to diagnose as leprosy, and we're not sure which is going on here, but the pathogens have picked up something else, which is quite interesting. And we are still diving into this a little bit more. But whereas a paleopathologist might have missed these things and mainly be able to say that this person looks healthy, the DNA evidence has shown us that something else is going on. The teeth also tell us that there's a bit of stress, and one of the biggest indicators that there is something happening with this person is that she didn't survive. We don't know why, but she died probably in her mid to late 20s. Okay, so it just kind of highlights that somebody who might look fairly healthy or at least not very sick, from a paleopathological perspective, there's actually more going on, and we can understand that better when we start to piece together these different forms of evidence. So this is kind of tied to an individual case, but on the broader case, it really is kind of what I try to do. It's look at different forms of evidence to get a more complete picture of what is happening with health. And so what we're seeing in Denmark is changing circumstances through the period being that I've studied. We have one thing that always affects results is we actually have way fewer people in cemeteries post Black Death. That could have to do with a large segment of the population being killed by Black Death. It means that it's harder to see what's going on from a statistical standpoint. We do see a bit of shift in survivorship from the early to the just post-Black Death period. People just post-Black Death seem to survive a bit longer. We are seeing some demographic changes, some population changes. There is a slow urbanization, but we're not seeing a rapid shift after Black Death to urbanization. Denmark actually takes a while. So a big change that we're seeing is a population difference, but urbanization is not a rapid change that we see, okay? So we're still trying to understand this. There's ongoing work being done in Denmark to try to piece together on a broad time scale what people might have experienced. And sometimes it's hard to actually hone into the details because these timescales are so big. More recently, I've been looking at a population in Austria. So work with the Natural History Museum in Vienna and colleagues there. We're starting to look at the Avar population from the 6th to the 9th centuries. And this is a population who's lived in the Pannonian Basin, including what's modern day Austria from this period. And the interesting thing for me here is that I can look at more social indicators because while the medieval Danes weren't buried with any grave goods or social differentiators, the people in Austria are. So one of the things I'm interested in looking at is any indications from the grave of different levels of social status insofar as graves can tell us that. So we'll be looking at health and stress and tying into the broader framework of work that's already been done with these populations to try to understand them a little bit better. And part of this will be looking at paleodemography, so improved sex estimation from things like amelogenin, also improved age estimation using a new technique for transition analysis, which is a new age estimation technique. We will be doing the growth and development work, including body metrics and the dental work. And I'm hoping to improve how we actually quantify those stress markers. This is looking at trying to actually measure the distance between paracomata, those growth lines in teeth. That's very early stages. We'll be looking at paleopathology and tying this all into the social dimensions of health and stress. So that is something that we hope to use to gain a lot of insight into this population as we go forward. So just to sum up, I look at different parts of the skeleton. My focus is on teeth and everything that I can get from teeth. Part of this is tied to the fact that some of the analyses I do is destructive. And so what I tend to do is spend a long time documenting teeth. I use 3D scanning, for example, and photography, and then I do surface microscopy, and we do set up each stage of the analysis so that we maximize the information we gain before we get to another level of destruction. And ultimately, it means that level of destruction takes a long time. So, so ideally by the end, a single tooth will have given us a lot of information. And I tie this into the broader evidence from the skeleton to try to understand the life course, what happened at different stages of life, and what did that actually mean in terms of people's survival and their lived experience. And I think that is it. Thank you for listening to me. I'm happy to answer any questions you might have.
Thank you, Dr. Gamble. It was amazing. Well, I'm a bit biased, but it was amazing to know and to think how much we can learn about our past using dental anthropology. So thank you.
My pleasure.
I think we have some questions. So let us see here. I don't know if Dr. Daniel is going to. Well, meanwhile, I want to ask for Dr. Pedro to join us. Então, representando Dr. Pedro da Glória, da Universidade Federal do Pará, que está com a gente hoje. Muito obrigada de novo, Dr. Pedro, pela sua presença.
Eu que agradeço. So, Julia, thanks for your lecture. It's very interesting because it shows how bioarchaeology integrates multiple knowledges from historical, genetics, archaeological, and biological, of course. So this crossroads, I think it's much about what you do as bioarchaeologists. And I have two small specific questions for you. One is related to your hypoplasias data. And if you can say that, what about the size of these hypoplasias? Do you see difference between females and males regarding the size? And if the relation between frequency and size of hypoplasias could explain this difference of survivorship? So this is one question. And another one is, we have a term called osteological paradox that was proposed in the 90s. And how do you relate your data with this concept? And if you think this could be applied to your data, or if you think sometimes it can be true, sometimes the bones express or are related to survivorship, or sometimes not. When you can use this concept, when you can't. These are my two questions, and thanks again for your lecture.
Thank you. Really good questions there. And you're right, bioarchaeology is very multifaceted, and I should add that I can't do what I do without collaboration, right? So I'm not an ancient DNA specialist, so my colleagues and collaborators are the ones who really are the brains there. And the proteomics as well, I collaborate with proteomics specialists and bioinformatics people at the University of Manitoba, and they're really, they are my guides on that front. So it really is very important in modern bioarchaeology, I think, to do that. And in terms of the severity, how big the defects are for dental enamel, that's a really good question. So the way that I've gotten at that so far is to try to quantify severity. So I have, depending on the amount of the crown and how pronounced the defect is, I will score it as mild, moderate, or severe. And I've looked at that in relation to sex, and there isn't really a clear difference between males and females there. That could indicate that my way of scoring it is not maybe good enough. Maybe I need to continue to work on that. It could mean that there isn't a difference on that level. So it's something that I think still needs more work. I wouldn't want to say that we know one way or the other with it yet. It can be really difficult. also to quantify the older people, because you can get a lot of wear on teeth, and that can affect how severe it looks if you're just doing a surface analysis. So I think that is a really good question, and I don't have a clear answer. So far, it's not suggesting that severity will have an effect, but I wouldn't want to commit to that at this stage. And for the osteological paradox, I think the individual who I showed from TIRIP is a really good example highlighting kind of the osteological paradox, because that is somebody who a paleontologist might have called healthy, for example. So it highlights on an individual level that a healthy skeleton isn't necessarily healthy. How do we define health? And if that person were an old person with a lot of skeletal pathology, would that mean that they are unhealthy, or were they more resilient and therefore survived longer? Because it does kind of highlight that the osteological paradox on an individual level. In terms of the dental enamel, when somebody experiences a stressor, we don't exactly understand what that means in terms of their health and resilience. Did the person who has more stress have more stress because they were more susceptible to stress or to forming stress markers in response to stress? Does it mean that they survived a little bit better, or were they in a context that was conducive to them experiencing stress and developing the markers? We really are not entirely sure. So we don't know whether it's something innate or something environmental, and we still have a very poor understanding of stress response in relation to likelihood of developing these defects. So with the osteological paradox, being able to look at a person with enamel hypoplasia or the internal markers and say that means they're sick is not really possible. And I think that is kind of a cautionary note of the osteological paradox, that kind of saying that we cannot say those things. We need to accept that there are multiple interpretations in terms of health or sickness. So it's a lot more complex. The survivorship indicators may point to continued circumstances that affected people both in childhood and in later life, or to that stress impact actually changing how they responded to stress. We're not entirely sure. We have to keep open to different possibilities. Does that answer your questions?
Thank you very much. Nice.
And I'm not sure that I'm seeing any other questions come up, Fernanda. I may not be looking in the right place.
Yes, Dr. Danilo is putting a question here. So I just want to thank you again for Dr. Pedro being here with us. And we have a few questions actually from Bruno. Just a second. Can you see it, Dr. Gamble?
Yes, I can. So the question is, why Denmark? Is there an interest in assessing other regions that were intensely affected by plague? So that's a really good question. The reason Denmark actually was because my PhD supervisor, who is Dr. Rob Hoppe, already had collaborations there. And so during my PhD, I went to Denmark. Now, another reason that collaboration exists, well, there are a few reasons, but one is that the ADBU, the anthropological department at the University of Southern Denmark, has a very large and important repository for human skeletal remains. So it's a really good place to look at medieval patterns in health. And I've always been interested in the medieval period. My master's did work actually a bit earlier, more consistent with the Avar population. I'll be studying, I'm studying now. So early medieval period, but I've always been interested in the medieval period. So part of it is just chance in terms of who you know and where they're working. Part of it is this interest in medieval period, and part is just the amazing collection at what's known as ADBU. So, and Dr. Jessica Boltson, who really was developed the lab there, has been for many years doing extensive work to kind of work on these populations and make it accessible to the international community.
Thank you. Okay, so I have a question. It's not really a question. And after mine, we have one more. And then I think we can finish. But, well, I want to go to this field in bioarchaeology and biological anthropology. And I'm just starting. So I wanted to ask you, what would you say for those who want to pursue a career in bioarchaeology and biological anthropology once we can see that they are really interdisciplinary?
So I think advice I would have would be to keep your doors open because they are so understanding past health requires a broad lens. So get a strong background across areas so you'll at least have some foundation, say in chemistry, biology, and to not be afraid to reach out to people working in the field and to ask questions. I think that's really important, and people are usually happy to talk about their research, and you can learn a lot from doing that.
Thank you. Would you say that the most difficult part studying this involves the samples, how hard it is to work with ancient DNA, and so, but first it's so hard to find, right? And second, it's all really complicated. You say that you have just one sample, right? And you have to kind of destroy it.
Yeah, so archaeological samples are far from perfect experimental samples, right? So there's always going to be limitations with samples, and part of what we do is trying to recognize those limitations and how different preservation or bias might influence results. So for me, for example, looking at teeth, one of the big limits I have is older people, their teeth get worn, and that can obliterate, get rid of information. Now, there are some large samples around the world, so large collections you can study. I think we need to be very careful in deciding to do destructive analysis. So for example, I did cut a number of teeth. I thin sectioned them after documenting very carefully during my PhD. And we've done the DNA work, which is destructive. But I haven't actually thin sectioned any more teeth since my PhD. And that's something that I will do very carefully if next time I think to do it, once all documentation and all evidence can be acquired from it beforehand. So I think if you can answer a question without destructive analysis, you should. A destructive analysis should be a last resort and very well justified. So that's why I take the approach I've taken, but you need to be very, very careful, I think, in how you go about that. Sample sizes will always be an issue. That's just unfortunately one of the things that we need to deal with.
Yes, thank you. Okay, here another question from Bruno.
So someone is asking here whether it's possible to assume that the burial sites are exclusive for people who were affected by plague. So in the case of Denmark, no, that we don't actually have plague pits for the Black Death episode anyways. There are some possible plague pits, but not associated clearly with the Black Death. It's not like England where you get Smithfield and where you get these plague pits that were actually pre-prepared as people saw that plague was coming. Denmark doesn't have that. So the populations we sampled, were not clear plague burials. And part of this was seeing on a broad scope what diseases were present, right? The initial focus of this study was not plague. It was the disease sphere, kind of what people could have suffered from during this period and experienced. Plague ended up being the first one we really honed in on, and we did find it in these general population cemeteries, but they were not targeted plague cemeteries.
Thank you. I think we have one more.
What is other information I'm researching? So I am also collaborating with folks who have more know-how in isotopes than I do. We're hoping to do some isotopic work connected to stress in teeth, so the ability to ideally map chemicals like strontium and barium that may have a connection to, for example, starvation has been proposed with release from the bones, which are a repository. If we can maybe see an indication of these in that parallel the growth markers or the stress markers specifically in teeth, we may be able to hone in a little bit more on the cause of those stress markers. So trying to basically combine multiple modes of visualizing stress markers. I've only done visual, but we'd like to do chemical. That's one area. And beyond that, I don't think ancient DNA, there may be further expansion. There's still a lot we want to do. It's part of this is also figuring out what you can do with the time that you have. I have dental calculus I would like to look at. I always save dental calculus if I clean teeth. So those are also aspects that we could look at for especially diet, and also dental microwear for diet. So there are different things to look at, but they're not kind of immediately things that we'll be doing.
Nice. We hope to see more of your research soon.
Thank you.
Okay, Dr. Gamble, if you want to say anything else, I think we are done with the questions.
Thank you very much for having me. It's been lovely to speak to you and to meet you. We might have one more question there. How do you control for overall life expectancy between males and females for your survival analysis? So that's a really good question. So we don't really know life expectancy here. This is simply based on the ages at death estimates tied in with the indicators of stress. And ages at death are far from perfect. With paleodemography, we're hoping to improve those estimates with the new transition analysis technique. But one of the things that that we're very bad at for bioarchaeology is estimating age at death. So it is definitely not perfect. Yeah, I hope that answers your question.
Thank you, Dr. Gamble, again. Okay, so please continue. People thinking.
So thank you. If anyone has any other questions, you're welcome to reach out to me. And I hope you've learned something. And it's been lovely to talk. Thank you so much for hosting and for organizing this. It's been wonderful to meet some new people.
Thank you so much, Dr. Gamble. Well, your email is here on the screen. So it was amazing to talk to you. We are really happy that you accepted our invitation. So we hope to talk again soon.
Thank you. Take care.
Ok, muito obrigada, pessoal. Obrigada, professor Danilo, mais uma vez. E encerramos por aqui, então, pessoal, nosso terceiro episódio do LEABTalks. Fiquem de olho nas redes sociais e no site do LEABTalks para acompanhar o que vem mais para frente nessa temporada. Obrigada, gente.